So I'll tell you a story today about anticipatory binding in phage. And this is a project we've been doing with Jeff Miller at UCLA, who's responsible for discovering the system. So the first question is, what is anticipatory binding? So anticipatory binding is a mechanism that biological systems use to adapt to a varying and unpredictable world. And the place we know it best from is our own adaptive immune system. So we don't know what new pathogen will emerge, and our strategy, and the strategy for vertebrates, is to make a repertoire of sequence diverse proteins. And we hope that in this repertoire there's going to be one protein, at least one, that will recognize some pathogen by just a Darwinian selective process. And that process then, of course, depends on a large number of variants. And we find that in our adaptive immune system, we dedicate something like a megabase of DNA for genetic recombination mechanisms to generate something like a massive number of potential antibody or T cell receptor sequences, something like 10 to the 14th or 10 to the 16th. So that's one place where we know that there's massive sequence variation that brings about this sort of anticipatory binding. The only other place we know of that does this is also in vertebrates, but jawless vertebrates, who have a separate branch of immunity that depends on a different protein fold from antibodies and T cell receptors, leucine-rich repeat proteins, doesn't really matter, but they also use kilobases of DNA in a different genetic mechanism, but still involving recombination of genes. So those are the only two places in biology that we've seen this strategy used, of massive variation and then selection from this repertoire. Until my colleague, Jeff Miller, discovered diversity-generating retroelements. So DGRs for short. And since his discovery originally in bordetellophages, which I'll tell you a little bit more about in a couple of seconds, DGRs have been identified in a wide variety of bacterial phyla that occupy diverse ecological niches, from warm tropical waters to Arctic permafrost. They're found in bacteria and phage, in bacteria that have planktonic lifestyles, consortial lifestyles that are symbiotic, pathogenic. And you'll see they're distributed both in bacterial genomes and also in blue, phage and plasmids. Now, how are DGRs identified? So there's different flavors of diversity-generating retroelements, but the phage element serves as a nice prototype. They're made up of a protein that gets diversified, that has a variable region at its C-terminus, and almost always at its C-terminus, of about 45 residues. And that's the only portion of the protein that gets diversified. That variable region is repeated downstream as a almost identical copy, about 90% identical template, called the template region. And in between, there's an accessory protein I'll tell you a little bit more about. And importantly, downstream of that is a reverse transcriptase that's got a unique sequence characteristic. And that element takes up of about only 3 kb. So that reverse transcriptase falls into its own special clade based on sequence. And you'll see based on its activity. So this is a figure that Mary Gingery in Jeff's lab came up with, and shows that the reverse transcriptase is related to other reverse transcriptases like LTR and retrovirus reverse transcriptases, but forms its own clade, and in fact is most related to group 2 intron reverse transcriptases. Now, based on the existence of this unique reverse transcriptase and these two direct repeat regions, one in an open reading frame, we've been able to go on, we meaning a couple of labs, including my own, have been gone on and identified DGRs in different places. Among these was actually in Rick Bushman's lab. Rick used the same kind of motifs and looked at DNA viruses found in the human gut and identified a number of phage that encode these kinds of elements, having variable regions and template regions, and also these variable proteins. So our gut are filled with the bacteria phage that have these elements. And more recently in work we did with Blair Paul at UC Santa Barbara, using metagenomic sequencing, he recently identified DGRs in a virus of archaea that likely infect methanotropic archaea found in subsurface sediments and methane seeps. And he's also identified them in nanoarchaeota, very strange archaea that live as parasites on other archaea. So how do DGRs diversify? And so this is a general model of how they come up with protein diversity. So the template region is transcribed into an RNA, and there's a complex that's formed between the accessory protein and the reverse transcriptase, then reverse transcribe this RNA into cDNA. And that cDNA somehow makes its way back into the coding locus. And that's a process we don't quite understand yet. And so this part is based on real speculation. We think at the locus, where there's this variable region, there's a nick that's made, and the RNA then acts as a template and is primed by the target itself. So this is target primed reverse transcription, and we get a cDNA that's made and homes to the locus, and then somehow replaces this locus, or this coding sequence. And importantly in this process, the reverse transcriptase has pretty good fidelity, except at adenines. At adenines, it randomizes the sequence and puts in any base. So as a result of this process, you get adenine-specific mutagenic retrohoming, where this sequence is now replaced by a new sequence where just the adenines are changed. And so the place we understand it the best, and the place it was characterized first, was in bacteriophage that infects the bacterium Bordetella. And this is work by Hong Zhao at UCLA, who determined the structure, the cryo-EM structure of the virus. And importantly, what we found is that the protein, that's the variable protein in this bacteriophage, is the receptor binding protein for the phage. So that's shown here. MTD is the variable protein and exists at the tip of these 6 tail fibers that come up from the phage. And in fact, we think there are actually 2 of these proteins at each fiber, so there are 12 total receptor binding proteins at the ends of the phage fibers. And so why does this phage need to vary? Well, it turns out to keep pace with its host. So Bordetella changes the kinds of proteins it has on its surface based on where it is. Bordetella, as you know, is a pathogen, and oops, going back. So when it's in the host, it expresses a set of surface proteins that are required for virulence based on a 2-component system called BVG. So this is the BVG plus state. And when it goes ex vivo, it turns off expression of these surface proteins, which could be potential receptors for the phage, and expresses new proteins on its surface. In fact, there's even an intermediate stage. So this phage, which is lysogenic, if it's gone into, let's say, the plus phase, and Bordetella meanwhile changes phase, it then has lost its receptor, so it's no longer functional. So as a strategy, it makes a certain number of variants, about 1 in 10 to the minus 6th, or 1 in 10 to the 6th, that then, by selection, will pick out a new receptor on this phase of the bacterium and infect. So a number of years ago, we determined the structure of the protein that's variable, and that's called the major tropism determinant, MTD. It's a complicated protein. It's a trimer. But what's important is that the variable region is at this very C-terminus of the protein in a C-type lectin domain. And if we look at that C-type lectin domain, we find that there are 12 residues that are variable because they contain adenines in their coding sequence. And all 12, shown here in the red, are surface exposed and form a binding site for the receptor on MTD. And what's really amazing about the system is that these 12 amino acids are so back up for a second. So these 12 amino acids are variable because they contain adenines in their codons. And if we look at the sequence of the template region, the residues that have symbols underneath them have adenines in their codons. And most of them are asparagines, so AAC codons. And by changing the first two A's of AAC, you can get to 14 other amino acids. You miss some, so for example, you don't get glutamate, but you get aspartate. You don't get lysine, but you do get arginine, which means that you get all of chemistry from this kind of system. And importantly, you get no stop codons. And now if you work through the combinatorics of how many different sequences you can get, this protein has, in principle, about 10 to the 13th different sequences it can encode. So what we've learned since then is that this platform that displays these residues is very stable. So this is a structure, an overlay of 5 different structures of the protein with 5 different receptor specificities. And you can see the backbone is almost exactly identical. So this presents basically a surface for the combinatorial display of variable residues to give you new receptor specificities. The other things we've discovered is that this is a conserved fold, in that we looked at other DGRs who have variable proteins whose sequences didn't suggest they were the same, but when we saw the structures, we found that they were exactly like C-type lectin domains. So this is one from a treponema denticola, which has only 16% identity to the Bordetella protein, but has exactly the same kind of structure. So it's a conserved fold for display of sequences. And amazingly enough, this protein has a potential of 10 to the 20th sequences, way surpassing the immune system. So in the last couple of minutes, I'm going to tell you about a short story about how we learned this phage works in terms of diversity. So this is a question that addresses how sequence diverse repertoires work. And it's a story that allowed us, because of the phage biology, to move from one specificity to the next. So we worked on a phage that infects through a receptor called protactin on BBG plus phase. That phage gave rise to a daughter phage through some sequence changes in this protein that infects the minus phase of the bacterium. We don't know what its receptor is, but we realized that this protein still bound protactin, but non-productively. And then this phage gave rise to one that reverted back to bind to the positive phase, called BPP6, back to the protactin receptor. So going from protactin to non-protactin involved 9 of the 12 residues changing. And what we've found is that this and this phage bind to protactin basically the same place, but not exactly. And when it reverts back to binding protactin, it binds to the same part that this one bound, but more tightly. Okay, so going back, it then changes 5 out of 12 residues. So we wondered, if this phage, or if this protein binds protactin, why doesn't it infect? Okay. So what we learned from trying to look at this example is that we saw the structure of the protein MTD bound to the receptor protactin and asked how well does it bind. And what we used was, to look at this, was a metric called surface complementarity. So that's asking how good the fit is between protactin and MTD. Perfect glove and hand fit is a score of about 1, or a score of exactly 1. And when you look at evolved interactions like oligomeric proteins, so evolved over time, they have a very good fit of about 0.8. But when you look at antibody-antigen interactions, these anticipatory binding events, they actually have pretty poor fits of about 0.64 to 0.68, because there's no evolution. They're happening through just selection. And when we looked at this interaction between protactin and MTD, we saw the same thing. There are actually 2 binding sites, but they both have pretty poor interfaces. Okay, so they have surface complementarities of down to about 0.6. So there's non-optimal complementarity, which suggests this interaction is happening by chance rather than through evolution. So we then went on to measure the affinities using surface plasmon resonance of these interactions. And what we found was that for the positive, for the ones that are biologically significant, they had pretty good binding constants, but not excellent binding constants. So they were down to about 3 micromolar for the positive, again 7 micromolar for the positive, and this one that still binds is about somewhere around a millimolar. So there's about a 100-fold difference. Okay, so that raised a question for us: does biology really work with 100-fold differences? That seemed to be pretty small. So what we knew too, of course, is that this is not the real case physiologically. You don't have a single protein binding to protactin. You have a phage. This phage has 6 tail fibers and up to 12 proteins. So we did the same experiment with phage and found the binding now went up to picomolar levels. So 8 picomolar, 9 picomolar, and this one also increased, the negative one also increased, but only to micromolar. So now between the positives and the negative, we now have about a millionfold difference, and that really does seem to be how biology works. We verified this by doing the same kind of experiment. So that's avidity, multivalency causing tighter binding. We did the same kind of experiment with phage and Bordetella and got very similar results, again, picomolar binding. So what's going on here? So again, just to show you what that data said is that this avidity interaction amplified the interaction strength. So we went from went from micromolar to picomolar, so about a millionfold jump, but it also did it differentially. So the negative one only went from about millimolar to micromolar. That was only a 10 to the third jump. So we made one much better than the other. And how did that work? So there's a way to look at this, and this now comes back to the immune system. And so if we've got a sequence-diverse repertoire, and we look at any kind of ligand, what we'll have is a distribution of proteins, most of which don't bind very well, and there's some exponential distribution, and we'll get, just by chance, some that bind very, very well, okay? And so what happens? Well, it turns out that in the immune system and in this system, the positive ones have pretty good binding, but not great binding. So surface complementarity is about 0.65, and KD's in the micromolar range. And that's true of primary antibodies and T-cell receptors. But on top of that, what you build in is avidity. So avidity basically acts on this and increases its binding strength by an exponential that's related to the multivalency. In our case, that was about 2. We went from micromolar to picomolar. And that put us in the range where we got an infection, a real biological effect. And that's true for the non-productive one, because that goes from about millimolar and also gets amplified by this factor of 2 to now only a micromolar. So again, by this exponential, we increase the difference between the productive and the non-productive interaction. And so that kind of increase, that factor of 2, is also seen in the immune system. The immune system, we've seen numbers of anywhere a little bit above 1 to about 2.5 for a pentameric IgM. So at heart, what's going on, we think, is that in a sequence-diverse repertoire, you can have a regime where you have high complementarity, let's say 0.8, where you choose only the ones that bind really well by themselves, and you won't have many of those. So that puts you out here where there are very few proteins that bind very, very well without amplification. Or you could have something that has lots of amplification, lots of different binding arms. And then that exponential factor is large. Oops, let's go back again. Exponential factor is large, but instead you then choose a lot of proteins that can bind, and you essentially create molecular glue where there's no selectivity at all, and possibly of self-aggregation, stuff that becomes just so sticky there's no selectivity. But it looks like that both systems have chosen an optimal balance of the two. You choose proteins that bind just okay, a surface complementarity of about 0.65, and you amplify that, but just a little bit, not too much, somewhere with an exponential of about 2, and then you get both efficiency, you have a fair number of proteins that are able to bind, and selectivity. Not everything can bind. So we think by studying this phage example, we've seen principles that are also used in the adaptive immune system to select out from a diverse repertoire and find ones that give you biological selectivity. Okay, and I've summed up work of a lot of people in my lab. This has been a great collaboration with Jeff Miller at UCLA and also a number of other people who've been involved in this work, including Blair Paul and David Valentine's lab. And I'll stop there and take any questions if there are any.